Dr. Shyam Prasad Sajankela has completed his Masters in Biosciences and PhD in Genetic Toxicology from Mangalore University, Karnataka, India. He has received postdoctoral fellowships from Lombardi Comprehensive Cancer Center, Georgetown University, Washington, D.C., USA, and Department of Radiology, Thomas Jefferson University, Philadelphia, USA. Dr. Sajankela has 18 years of professional experience in academics. He has published over 18 papers in various reputed science journals, both national and international. He has published several science articles in various magazines in English and Kannada languages. He has presented papers at various international conferences and also delivered several talks and lectures. Dr. Sajankela has received several grants, including the VGST grant, Government of Karnataka, and KSCST grant, Government of Karnataka. He has been bestowed with several awards, including Best Teacher Award 2012 at Manipal Life Sciences Center, Manipal University, Junior and Senior Research Fellowship for Doctoral Degree at Mangalore University, and Best Science Kannada Article 1998 by Karnataka Sahitya Academy. Dr. Sajankela is currently working as Associate Professor, Department of Biotechnology Engineering at NMAM Institute of Technology, Nitte, India. I welcome Dr. Sajankela to this session. Over to you, sir.
Thank you. Thank you for those generous introduction. I hope the PPT is visible. It's visible. So, when I was asked to talk on this particular event, and I was told that with students in mind, to come up with the topic which is very basic, which can easily be conveyed to students. So this is the topic which I have chosen. It's a very basic, not much technical. I may be kind of disappointing the experts, but this, as I said, it is directed mostly towards the students. So with that pretext, let me begin. Yeah. So what do we have common with stonewash genes, home pregnancy test kit, with bread, cheese, and wine? Obviously, since we are talking about biotechnology, all of us can guess the answer for this one, right? All of these are examples for manipulating organisms to create products or new processes. Basically, they're all the products of biotechnology. And for those who are wondering, like, where does stonewashed genes come in here? As the name indicates, stonewashed genes. It used to be the norms earlier, but nowadays, to avoid the practical issues, people have come up with cellulases to get the same effect like a stonewashed genes. So again, we have the connection there back to biotechnology. From age-old fermentation process to gene editing and beyond, the biotechnology has had its impact on our lives through various different ways, whether it is in the form of crop improvement or improvement of nutrition values, harnessing the microbes, or unique capabilities of microbes, extremophiles, in diagnosis of diseases or disease prevention, whichever field we are talking about or whichever concept and area we are talking about, the biotechnology has had its tremendous impact on all these. If you look at the history of biotechnology, if you just give me a second, I was told my voice is breaking. Is it okay now? Is it fine now? It's fine? Probably, I think it's a network issue there. Yeah, check it out. Yeah, sorry for the interruption. So if we look at the history of biotechnology, starting from fermentation to genetics, vaccines, DNA structure, monoclonal antibodies, PCR, transgenics, cloning, human genome projects, then genome editing, all those we have, seen the growth of biotechnology tremendously. But it is kind of difficult to predict as to where we will end up. I remember recently, like last week, reading an article where it said it's from an authentic source, where they're saying that in certain countries, people are working on certain elements which can alter the gene expression in a human being, especially humans are the target, wherein the efficacy of the person can be either diminished or enhanced, whether it is the performance with respect to whatever skills or his ability to think, creative thinking, execute the functions and execute the ideas, all of this. So it always gives us kind of, a serious thought that where are we heading? If it is in a good line, yes, we are okay. But if it falls into the other side of the line, then it's a matter to worry about. Having said that, if we look at the concept of biotechnology or development of biotechnology per se, biotechnology is a kind of tree which is developed from the inputs from various different fields, whether it's chemical engineering or cellular and molecular biology, or talk for that matter, immunology, biochemistry, genetics, human, animal, and plant physiology, and many, many, many more different fields act as a feeder for the flourishing of the field of biotechnology. But what are the implications of that? We always have this particular, concept, the query in mind, like why and where and how does this biotechnology will take us, and what all different aspects it has touched upon. Knowingly or unknowingly, or intentionally or unintentionally, all these different feeder cells from or different streams has converged into biotechnology, which has had effect on, let us say, energy and environment management, or plant agriculture improvement, animal agriculture, fermentation technology, food innovation and processing, whether it is to increase the nutritive value or shelf life or the other aspects of the fruits and vegetables, or to have a plant which is disease resistant, frost resistant, or all those kind of things, to diagnosis and healthcare, pharmaceuticals. But these fields are not all. There are much more different areas which are connected and associated with biotechnology, either as a feeder or as a downstream effector streams. The next slide, this particular slide will give a little enhanced version where you can see different areas such as human anatomy or genetics, molecular biology, immunology, mathematics, physics. All of them will contributing to the flourishing of biotechnology, and pharmacogenomics, biopharmaceuticals, nanobiotech, all these are some of the areas which are having the downstream effect as well as in terms of development. Now, when we look at the life form, we all know that the importance of DNA in life, in the sense DNA is a molecule, master molecule, who harbors all the information which is required to form an organism. When I say to form an organism, it is something like, a mango tree acts like and functions like one, because the information that is required to make a mango tree is present in DNA. All right, all the genes which are required are present in the DNA. So DNA will tell you what can happen, in the sense, based on the information whatever is stored in the DNA, one can tell like what is possible out of it. And if you are studying the factors associated or phenomena associated with DNA, you call it as genomics, and it is predicted that roughly around 20,000 genes are present in human beings. The next level or next level in the flow of information is the RNA molecule. So this RNA molecule gives us the insight into what appears to be happening, in the sense whatever information was present on the DNA, now it has got converted into an RNA. So that means to say that gene expression has started. So it gives a kind of snapshot into the cell or a organism's tissue or whichever it is, as to what appears to be happening. Now, if you look at, or if you study the concept of RNA and the phenomena associated with that, you term it as transcriptomics, and roughly around estimated 100,000 transcripts are present. But now the numbers doesn't match there. When you talk about genes, you say that 20,000. But when you come to transcriptomics, you say that it is 100,000. This is all because of splicing and splice variants and those factors contributing to the so-called discrepancy in the number. Now, you had information in DNA that got converted into RNA. Now, the language of nucleic acid has got converted into a language of protein. So, when you're looking at protein, the proteins will make it happen in the cell. In the sense, whatever information was there in the DNA, it's fine. But if you want that information to be executed, you need to have the protein. So when it comes to protein, you call it as proteomics, and the number even shoots up by one more zero there. All those put together, or from protein, you will be like looking into a metabolite of a cell or a phenomena, which will tell you what has happened in the cell or what is happening in the cell. And again, if you look at the metabolomics number, it's still there is a probably it is in kind of not full-fledged with respect to evolution, development, I guess. We still are to look into more detail as to the diversity of metabolites and other things. So all those factors, whether it is DNA, RNA, protein, and metabolite, all of them put together will bring about the phenotype. If you are talking about the color of a plant or a height of a plant or the fragrance of a flower, whatever is the phenotype you're talking about, the information, all that is required by this plant will be present, which will be executed in the above mentioned pattern or format. In biotechnology, we are always looking into genes. One of the possibilities, looking into genes, try to manipulate it, play around with that for the betterment of human race, whether it is our lifestyle or agriculture, whichever field you're talking about. We will be playing around and with the genes. So there are numerous tools and techniques which are available and made use to study the processes involving DNA, RNA, proteins, and their interactions. Okay, how DNA is playing role, how RNA is playing role, or what proteins are doing, and what is the combination or interplay between the different entities. That to study that, numerous tools and techniques are available. For example, to manipulate the DNA, you will have the molecular scissors, that is restriction enzymes. Then you have sutures, which is ligases. So different DNA modifying enzymes are present. So at each level, they have their own set of tools, techniques which are available, so that we can play around with that or modify it according to our needs. When we say manipulating the genes, it not only involves the manipulation of just a gene, like you bring in a gene which confers resistance to a particular disease or enhances the quality of a plant or whatever it is. You also at times will be playing around with or altering the regulatory elements of the gene. So is one of the routine technique in biotechnology, whether you are manipulating the gene or whether you are altering and manipulating the regulatory elements. So this is one of the routine techniques. Now, we have done some modification. Now, I don't know whether my modification, does it have any effect? Does it bring or has it transformed into what I intended to? That is a question. So I need to study that, whether my modification has been successful or not. So to do that, to understand the effect of such manipulations and modifications, the measurement of gene activity is kind of essential, all right? And some of the most common approaches are RNA-based approaches, then protein-based approaches. Then you have such as Western blot, reporter gene assay, chip assay. So different approaches are there. So now onwards, I will be concentrating on two different concepts which play or which contribute to the this particular biotechnology related biomedical field. First is, I'll be talking about reporter gene assay. As I said, when we manipulate a gene, we need to know whether it is effective or not. So to tackle that or to understand that, we will be one of the classically and one of the routinely performed assay is our test is the reporter gene assay. So these genetic reporters are commonly used in cell biology in order to study the genes, their expression, and other cellular events which are coupled with gene expression, such as receptor activity, intracellular signal transduction, mRNA processing, be it protein folding or protein-protein interaction, whatever the downstream effect is. But you need to study the expression of the gene, whether it has happened or not. As kids, we were all fascinated by these fireflies, right? All of us know that this concept of bioluminescence. These luciferases are oxidative enzymes that are able to or enable the organism that express those molecules to bioluminesce or to emit light. This is one of the most commonly famous and commonly used enzyme, which is firefly luciferase. You have a luciferin molecule which combines with ATP and oxygen in the presence of oxygen, and the luciferase enzyme will convert it into oxyluciferin along with emitting the light. So basically, the fireflies are capable to emit light via a chemical reaction, which in which luciferin is converted into oxyluciferin by the enzyme luciferase. Now, one thing I would like to mention here is that whatever we observe in nature may be with respect to a different context, but the current day scientists or creative thinkers have tried to use a totally different context into a method or an outcome which I want to analyze or I want to study. So how do we connect these two? Basically, luciferase or this bioluminescence activity can be used to study the gene expression. Very briefly, we will look into that. Okay. The essence or kind of principle of this whole approach is you have a protein of interest which you want to study. All right. So when you want to study that, you will generate a vector which has a promoter region into which the luciferase reporter is attached. Okay. Now, if your protein of interest is active, or if your protein of interest is able to bring about activation of gene expression, then the luciferase will be produced, and that luciferase will form the bioluminescence, or it will start giving out light. This is a simple assay. But now the same concept can be made use to study various different aspects. Now, what do I mean by that? Suppose I want to know that the promoter which I'm looking at, promoter for a particular gene which I'm looking at, whether which is the core area of my promoter? From what length to what length is my promoter area? That kind of study can be performed by this luciferase assay. So to perform that, what you will be doing is you'll be cutting some portion of that promoter region, fusing it with the vector PGL4 or most commonly used, then perform the same experiment. That is, put into the cell, look for the luminescence, measure the luciferase activity and intensity of signal that you will measure. But compared to the first one or the full length promoter which you had initially chosen, you find that when you cut down certain regions of the promoter, you will find that the activity has reduced significantly. Or another possibility is you will find that there is no expression at all. That means to say that that entire length of the promoter is essential for the gene expression. But if the expression has reduced by 50% or so, you can assume that, okay, still even if I cut down certain segment of the promoter, my gene is still active, or my gene can still get expressed. So that means to say that, okay, that promoter is still working. Cut down further. Again, do the same exercise, put into the cells, look at the luciferase activity, and then you will be able to know that whether my promoter, or which is the full length essential length of the promoter, or what happens when you cut down sequentially segment by segment of the promoter. This is what the luciferase assay can give insight into. Or you have another molecule which is a repressor of transcription or gene expression. Put into the system, expose cells to such chemicals or such biomolecules, you will find that there is no activity or reduced activity. So which again can give you an idea as to the efficacy of that particular molecule. So basically, whether protein of interest or a molecule is an activator or a repressor, be very efficiently studied using this particular approach. Not just that, there are certain genes who has their promoters which is bidirectional in nature. Okay, now even this kind of activity can be studied in the sense on either direction. When they get transcribed, two different products are formed. So how do I study that? So to look into this kind of phenomena, you will have a dual luciferase system, wherein one of the product is firefly luciferase. The second one is renilla. It's derived from sea pansy. Okay, and on one direction onto which the transcription factors will be binding. So by using wild type versus stepwise mutating certain core regions, you will have the promoters which are essential for the transcription to occur. So using one simple assay, the outcome can be genuinely numerous. Various different aspects can be studied using the same assay, but it is up to you to decide as to what do you want out of that particular analysis. So some of the applications of this luciferase reporter assays are gene expression, obviously gene expression analysis, promoter structure analysis as we discussed just now, then SNP analysis, then antiviral research and therapy. So these are some of the representative applications of this luciferase assay. Having said that, this is not the complete and exhaustive list. One need to keep that in mind. Now, so next I will be switching on to a different field or different aspect, which is radiopharmaceuticals. The moment we say radioisotopes, I think we will have that kind of mindset, that or fear kind of mindset. Isotopes, harmful, all those things. But I think if we had not used radioisotopes in biology, biotechnology, 50s. Or if you are looking to get broken down, what way it is down, all those things. So these breaking. Am I audible? I'm getting to hear that my voice is breaking. So these radiopharmaceuticals are isotopes which are bound to biological molecules to target specific organs, tissues, or cells within the human body. That's a kind of ultimate goal of using radioisotopes. You will be clubbing it with something else, some biological molecule, so that it reaches the specific target, okay, in the human body or within the animal. This is used for diagnosis, and increasingly currently it's being used for therapeutic purposes. That is target specific delivery of therapeutic agents or contrasting agents, whichever it is. So the number of radiopharmaceuticals in clinical use is growing rapidly. Voice is breaking still. Thus allowing the medical community to better access to detailed information. Now, when you get a clear picture of a patient, clear picture in the sense with respect to a disease, or the size of a blood clot in a person's body, the location of it, the magnitude of it, so that can help the medical community or the doctors to come up with a much better, well-structured, therapeutic regime. When we use radioisotopes for specific organs or specific types of tissues, you need to capture the image, analyze the image, and come up with a clear-cut idea as to where and this is brought about by a instrument called as gamma camera, which captures the images of organs or tissues of interest, and that particular process is called as scintigraphy. Now, as I said, if you want to target a particular organ, then there are certain requisites. I mean, I just cannot assume that if I inject this particular radioisotope, it's going to go to the cancer cell specifically or to the liver or something like that. I just cannot assume that. There has to be a specific mechanism. There has to be a specific approach. There has to be a specific approach. So to do that, you need to, you're targeting, let us imagine, which is expressed in the cell, on the cell, so which is the target protein. Now, develop a particular molecule, targeting molecule, whether it is peptide or a protein or antibody, whichever it is, okay? So which will specifically bind to only the cancer cells or cancerous cells. And you have to have a linker molecule which will enable your targeting molecule to be bound to a contrasting agent, aging agent, which is radioisotope in this case, for either PRET or CT, basically imaging or therapy. And your targeting, which will connect the protein or carrier to the radioisotope. So the length, flexibility, hydrophilicity, and overall charge, these are some of the factors which need to be taken into account when we are coming up with the linker molecule. The carriers can be antibody, proteins, it could be peptides, it could be small molecules, any of that category be the part of carrier molecule. Coming on to the target, it can be cell receptors, enzymes, transporters, transcription factors, or any of those any one of those can be your target molecule. So you will be planning and you will be executing according to your target according to your target. Now, when we are using radioisotopes, it can be used in various different ways. In the sense, free radioisotope, like with a small molecule, or you can bind it with the peptide or antibody, or you can have the radioisotope in nanostructure, or the entire microsphere which is loaded with radioisotope. Any one of the approaches can be possible. I think most commonly used ones are after the first one. The first one may not be that greatly used for specific purposes, because specificity may not be that when you use isotopes alone. Now, when we are, or when the radiopharmaceuticals are being designed, there are certain challenges one need to look into. First is production. The radionuclide, once you produce the radioisotopes, you need to incorporate into your target molecule or your carrier molecule. But that is not just enough, because once you incorporate that, there is possibility that radioisotope, whatever you have had, there are a lot of free radioisotopes. So basically, you need to purify them and the entire setup so that free radioisotopes are eliminated. Otherwise, what can happen is they can non-specifically travel to various possible organs, and it can give you a false image, false positive image. Then the quality control is quality control, such as chemical purity, radiochemical purity, then radionucleidic purity, pH, residual solvents, filter integrity, out of which pH, then residual solvents are certain very critical issues, because if when you're dealing with the proteins for targeting and your pH is messed up, then your entire setup can be dismantled, and you will have just the carrier molecule without the radioisotope attached onto that. This is possible. Then conditional release. There's another factor which needs to be looked into. Then administration to the patient or your test organism. If you are in a process of developing a product, then you need to do studies in animals. So how do I administer it? Oral, or is it intramuscular, intraperitoneal? Some of the cases, in mice, in certain experiments, tail vein injections are required. Just imagine you are handling a mice and you are to inject these radio-labeled product into the tail of the mice. So precision you can think of. And if you happen to have black mice, then the situation will be still worse. Quality control, that is post-release, that is microbiological purity and endotoxin level, is another point of concern which needs to be looked into carefully. And finally, the final release, after all these hurdles and initial productions, purification, looking at the quality control, conditional release, administration, and everything, does my molecule get released, and released in a proper way, in a proper dose? So these are certain challenges with respect to designing and administering and the effectiveness of the isotopes. So with radio pharmaceuticals in background, we will be looking into just a couple of examples of how radioisotopes can be used to image certain conditions. The first one what we're going to look at is deep venous thrombosis / pulmonary embolism. This deep venous thrombosis and pulmonary embolism are underdiagnosed but serious conditions. However, it can be preventable, in the sense, if it is diagnosed properly, if it is diagnosed well in advance and in a right time, serious consequences can be averted. Now let's understand what is this deep venous thrombosis. DVT, which is it's commonly called as DVT, or deep venous thrombosis. It occurs when the blood clot forms in a deep vein, and usually it develops in the lower leg, thigh, or pelvis, but they can also occur in the arm. Some of the symptoms includes swelling, excessive pain, localized pain, redness at the site. So these are some of the symptoms of DVT. But if you look in general, it looks like a very, very common kind of set of symptoms. The second condition is pulmonary embolism. This is the blockage in one of the pulmonary arteries of the lungs. Generally, what happens is the deep venous thrombosis. Once it happens, the possibility that the clot can break away from the parent block, travel in the blood, lodge itself in the pulmonary arteries, leading to severe consequences. Some of the symptoms include, or the symptoms, it depends on how much of the lung is involved in the embolism, then the size of the clots. Some of the symptoms associated with the pulmonary embolism are shortness of breath, chest pain, cough, then rapid or irregular heartbeat. So these combined or put together in a combined fashion can give an idea to an expert as to what has happened to the particular person or what is the issue with the particular person. So if these are undetected and left untreated, it can lead to serious consequences. So intervention is required. So what is the way to intervene that? The first foremost, visualize that, to see that a certain phenomenon, such and such phenomena has happened. Coming on to the imaging, first thing is like you need to image it. One of the commonly used agent is a pentapeptide, which has very high affinity for alpha chain of the fibrin. And when you label it with a radioisotope called as technetium-99, this is a radioisotope who has half-life of about 7.5 to 8 hours. Okay. When we choose the type of radioisotope for whether it is imaging or therapy or whichever purpose, the choice of radioisotope becomes very critical, because if you use, let us say, iodine or some of the radioisotopes whose half-life is thousands of years, then it resides in your body and will be like you'll be continuously exposed to those kind of radioisotopes, which is not advisable at all. So the best thing would be moderate energy and the half-life is reasonable, few hours is the best candidate for. So this experiment was done in pigs using the blood which is drawn from the pig.
Because that particular subject is added to the supplementary list actually. So if
rather than allow it to clot, before clotting, mix it with the tantalum. Tantalum is a density provider, and it is very tough material which doesn't get hydrolyzed by or degraded by any kind of
health reason, genuine, or you.
Once you generate the clot, or once you have the clot,
yes,
so whenever there is an examination, payment,
you will introduce the clot through the jugular vein. So technetium-99, which is used to radiolabel the pentapeptide, is injected, and the animal is imaged sequentially at different time points. It will be imaged. This particular image is of the pig, which is lying down or made to lie on its back. Okay, here on the top region you see some white area. This indicates the hot spot in the pig. This is actually the point where the radiolabeled pentapeptide is injected. This is the yellow. And if you look at this particular area where the arrow mark is there, you can start seeing the accumulation of radioisotope. You can see the presence of a significant sized spot as the time passes, and a number of spots are increasing. And in the neck region, you see a spot, there's a hot spot here. That is the area where intubation is given. That is attracting the peptides also. So finally you can see multiple different spots which is appearing as the time passes. One more thing I would like to bring your attention or draw your attention to. If you look at the first image versus the last image, in the first image, the density of those white spots are very intense. This is nothing radioisotope which is giving out a signal, and it is from the blood. But with the passing time, the radioisotopes from the blood gets cleared, and you can see the density or the abundance of radioisotopes is going down gradually. When you have imaged the pig for duration of 24 hours, close to 24 hours, you will euthanize it, and you will hunt for the spots which are yet in the gamma camera or through the gamma camera. So this spot is from the heart and lung area. So the best thing, I mean, under these conditions, visualizing minor clots is not possible because of the body density and blood and other things. It is not possible. So the best thing to do is excise out the lungs here, lungs and heart. This image gives you the picture of heart and lung being excised out. On the right hand side, you have a radiogram, and on the left hand side, scintigram, which is taken using the gamma camera. So in the scintigram, you can observe that there are numerous hot spots present throughout the lung, not much in the heart. Whereas the second image is of the corresponding same heart and lung area, I mean lungs basically, but you can hardly find only two major spots in the second image. So it is the issue with the resolution and those aspects. Once you get to this level, you will go to a specific region, excise out the area, and try to recover the clot, quantify it for further analysis. Okay, so this is how you will be using radioisotopes to image the deep venous thrombosis, or basically in common terms, blood clotting. Next, coming on to imaging cancer, this is a small segment. As all of us know that earlier the better with respect to cancer, in the sense earlier you detect it, it is always better, because chances of the patient's recovery, survival is all dependent on how early you can detect it and detect it. So early and reliable diagnosis of colorectal cancer continues to be demanding, and it is challenging. And different cancer cells express vasointestinal peptide, one of the specific cancer specific molecule or receptors in high density, such as colorectal and breast cancer will have this particular component overexpressed. So this concept is made use in diagnostic therapeutic approaches of the cancer. So the carrier molecules or system is loaded with the imaging or therapeutic agents with surface ligands to vasointestinal peptide, potentially to be actively targeted towards the cancer of interest which you want to image them. So in this image, the first image is, this image is taken using different radioisotopes and different agents or targeting molecule. In the first case, the first image is of the breast cancer, which is injected into the flank of the mice and nude mice receiving technetium-99 radio-labeled TP3654, the first image. The middle one is another mice, but which is injected with technetium 99 labeled TP3982, another peptide. Last one is done using the same peptide, but with the copper. So you can see that different peptides have the different penetrance level. How do I say that? Like if you look at the first and second image, in the second one, at the center, you can see that the area is slightly yellowish. Flame color is there. That's an indication that the level of, the area is more hot compared to the rest of the areas, or basically like more isotope has entered that particular area. So this way you can image the oncological disorder. So basically radioisotopes can have multiple applications. Okay, so these two examples. One is of reporter gene assay as well as the radionuclides, radioisotope in imaging are the two concepts which I chose, because it is humanly impossible to provide justice to cover entire array of techniques and tools which is used by biotechnology in biomedical field. So the reason why I chose is like restriction enzyme and certain bioinformatics, CRISPR. Some of the approaches are studied by the students. That's the reason I have not touched upon. So the area where it is not dealt in little or where they do not have much exposure, those areas I have touched upon at a very basic level, at the student level or student understandable level. So coming on to the conclusion, the biotechnological tools and techniques have boundless possibilities in diagnosis and therapy. I mean, sky is the limit, and your imagination is the limit, we can say. Example, advances in DNA technology are the keys to pharmacogenomics and personalized medicine. Here, I would like to, last two words I have intentionally highlighted in yellow, because people are working on the genomics based on Ayurvedic concept. In the sense, we all know that Vata, Pitta, Kapha is the concept of Ayurveda, and that is basically nothing but the body nature of a person, right? So are we just based on the particular person's body nature, is it characterized, or at the genomic level, are we seeing any difference between the people who belong to different Vata, Pitta and Kapha, and can we come up with a much more better, much more personalized medicine? This is the kind of work which is going on. And finally, these developments promise to result in more effective, individualized healthcare and advances in preventive medicine. I think with that, I would be concluding. Thank you very much. If you have, students, any questions, please, I will try to answer.
Thank you, sir, for this thought-provoking and informative presentation on using various molecules like luciferase and radioisotopes for development of pharmaceuticals and for making us aware of techniques like imaging tumors. Thank you once again for your awe-inspiring presentation. The session is now open for discussion. I request the audience to ask questions, if any.
No queries.
Up next, we have a talk on biomedical research, the road forward, by Dr. Radha Madhyastha, Assistant Professor at the Department of Cardiovascular Physiology, Faculty of Medicine, University of Miyazaki, Japan. I request Ms. Srividya to briefly introduce our guest speaker. Over to you, Srividya.
Radha Madhyastha is currently working as an assistant professor in the Department of Cardiovascular Physiology, Faculty of Medicine, University of Miyazaki, Japan. Her main research focus is on elucidating the molecular machinery involved in the ignition of chronic inflammation and the efficacy of nutraceuticals in elevating the same, especially in chronic non-healing wounds. She is currently working on the role of exosomal microRNAs as intracellular vehicles of inflammatory cargo. Her research career began with an MPhil degree in zoology from the University of Madras, followed by the PhD degree in zoology from the same university. During her PhD tenure, she was invited to Japan as a recipient of Japanese government Monbusho invited researcher. Her entry into the realm of molecular biology began when she joined the Department of Applied Physiology, Miyazaki Medical College, Miyazaki, Japan, as an invited researcher and then enrolled into the PhD course. In 2004, she received her PhD degree in medicine from University of Miyazaki. Since then, she has been working in the same department, guiding research students and pursuing her research interest. As of date, she has co-authored 40 research papers published in international journals, and has presented her research findings in 37 international conferences. In 2016, she was awarded with Women Researcher Encouragement Award by the University of Miyazaki. In the social arena, she works towards encouraging young women researchers to pursue their aspirations as scientists, researchers. Under the umbrella of Sakura Science Plan, sponsored by Japan Science and Technology, to date, she has facilitated to visit of about 50 young women researchers from India for training and motivation program in Japan. Over to you, ma'am.
Thank you for the introduction, and thank you, Dr. Kamath, for organizing such a nice interaction with the students. So I'll start with my presentation. Where is it? Share. Okay. Just give me some time. Share. Okay. Okay. Good. Today I'll be talking about biomedical research, my road, and the road forward. So I will not go into the details of my university because you already by now you should be knowing about University of Miyazaki. From morning you must have heard a lot about University of Miyazaki. So I'll directly go to my topic. Okay, so let me start with this picture. Can anyone tell what this picture is? I don't think many of you will be knowing what it is. I'll come back to this later. Please remember this slide. So first, let me start with my a little bit information about myself. Already, it's been although it has been I've been introduced. I'll just tell you I'm a molecular biologist working on basic medical research, and I prefer to work on cells.
Excuse me, ma'am.
Inflammation is my
Sorry for the interruption, but your screen is not on full screen mode.
Really? Okay, so I'll just. Is it now?
Yes, ma'am.
Okay. So my work is on inflammation, cellular inflammation, and I'm a molecular biologist. So and my interest, the center is inflammation, and the four pillars would be microRNA, exosomes, nanoparticles, and therapeutics. Why inflammation? Inflammation is the doorway for any disease. Actually, inflammation is a necessary response of the body. Okay, it's inflammation is necessary to tackle with any infection or tissue injury. If the inflammation process, our body's inflammatory response works fine, then we have the tissue repair response. Sometimes the inflammatory response goes crazy, becomes like it creates a havoc, and it becomes chronic instead of healing, and then we have a shift in the hemostatic shift in the set points and development of disease. So basically we can say chronic inflammation is the doorway for disease, pathological conditions. And then microRNA. MicroRNAs are very short, non-coding single-stranded RNA molecules, and they do not become, they are not translated into proteins, but they have very important role as gene regulators. Previously, they were thought to be just debris, but now they are known as master molecules or gene regulator molecules, and they're also now being targeted as biomarkers or target drug targets in these regenerative diseases and any pathological conditions. MicroRNA, we can see they have a role to play. So here we have the difference between mRNA and miRNA. miRNA, microRNA is called as miRNA. So in the case of mRNA, first we have the DNA. DNA is transcribed into RNA, and RNA is mRNA. mRNA is translated into proteins. But in the case of microRNAs, the same DNA is transcribed into what we call as primary miRNA. The primary miRNA is cleaved into precursor miRNA, and the precursor miRNA is further cleaved into small nucleotide sequences, which we call as mature miRNA. This mature miRNA does not get translated into proteins, but still it has a very major role. So here is the pictorial representation. First we have the gene. The enzyme RNA polymerase 2 will will act upon the gene, and will transcribe the gene into primary miRNA of about 400 nucleotides. This is as good as a full-length mRNA sequence. But this primary miRNA is further cleaved by an enzyme called as DROSHA into a 70 nucleotide hairpin structure called as pre-miRNA, precursor miRNA. So this process takes place in the nucleus, inside the nucleus. The precursor miRNA is transported by exportin 5 from the nucleus into the cytoplasm. And once in the cytoplasm, the precursor miRNA is acted upon by an enzyme called as dicer. It cleaves the precursor miRNA from 70 nucleotide into a 20 nucleotide mature miRNA. This mature miRNA forms a complex with a set of proteins, AGO1, TRBP, and the dicer itself to form a complex called as RNA-induced silencing complex. And in this complex, what happens is when any sequence, like there are some target mRNAs which have complementary sequence to this miRNA sequence. So the complex will bind to such target mRNAs, to the 3' UTR of the target mRNAs, and will either degrade the target mRNAs or will prevent its protein translation into proteins. So basically, if the target mRNAs of the miRNAs cannot be translated into the proteins. So my interest in microRNA started when I was working on diabetic wound healing. So this is a golden colored mouse. It's the KKAY strain. It's a diabetic mouse. It's obese and it exhibits impaired wound healing. So what we did was we used normal mouse and these diabetic mouse strains. We made full thickness wounds on the dorsal skin and we monitored the healing. So here we find that in the skin of the diabetic mouse, when compared with the normal mouse, we analyzed a panel of about 96 microRNAs, and we found that there were some microRNAs which were dysregulated, like 30-fold or 5-fold, 10-fold increase in comparison to the skin of normal mice. So this is the unwounded skin. Then we created wounds on the dorsal skin of the mouse. This white is the normal mouse and this golden is the diabetic mouse. So day 0, we made 5 millimeter punch wounds. The size is the same in both the mice. On day 4, you see that the wound is not raw. It is like sort of getting healed in the normal mice. And in day 8, it's almost healed. And this is the close-up of the wound during day 0, day 4, and day 8. But in the diabetic mouse, as you can see here, even on day 4, the wound is still raw. You can see this, there's still bleeding here. And on day 8, it's almost as equal. The size is almost as equal to the day 4 of normal mouse. So that means like it's about 50% impairment of wound healing in the diabetic mouse. So this is the H&E staining, the histochemical staining of the wounded tissues. The top portion, top is the top panel is the normal mouse, and the bottom panel is the diabetic mouse. This is left side we have the day 0 pictures, and the right side is the day 8 pictures. So you see there's a wound here created, and the same side of wound is created on in the diabetic mouse. But on day 8, normal mouse, the epithelium is almost healed. The wound is almost healed, but in the diabetic mouse, we still have a raw wound. It's not healed. So we profile the set of microRNAs to see whether there's any change in the wound healing pattern. And okay, so the first 3 bars here belong to the normal mouse data. The next 3 bars belong to the diabetic mouse data. The first bar is day 0, second is day 4, third is day 8 of normal mouse. Same way day 0, day 4, and day 8 of diabetic mouse. So we found that there are a set of microRNAs which have different regulatory, the amount of expression varied between the normal and the diabetic mouse. And I was particularly interested in this microRNA MIR21 because the pattern, like in all the other miRNAs, the pattern, the pattern was almost the same. Like there's an increase here, even in the diabetic mouse, there's an increase, but just the amount of expression was different. But in the case of microRNA MIR21, there's a complete different pattern between the normal and the diabetic mouse. So I was particularly interested in MIR21, and I started my further experiments, studies on MIR21. So this is the sequence of the mature MIR21. It's about 21 nucleotide sequence, and this sequence which is shown in the box is the sequence which is just important for the target mRNAs. You have this UAG, CUU, AUC. And if any RNA which has a complementary sequence to this in the 3-UTR, it is called as a target miRNA21, target RNA of MIR21. So if you have like complementary means the MIR21 sequence is UAG, CUU, AU. So the target would be having the opposite, AUC, GAA, UA. And we have about 3,000 RNAs which have this sequence. Like with some of them will have a slight difference, but it doesn't matter. It need not be 100% complementary. If it is 100% complementary, it would be mRNA degradation. The mRNA will get degraded. But if there is about 80% complementary or so, then the RNA will not be degraded, but the protein translation will be inhibited. So these are the few examples of some of the target RNAs, mRNAs of target. We can say target proteins because the mRNA cannot be translated into these proteins. And most of these, or almost all of these proteins or genes, are related to important physiological functions like proliferation, apoptosis, blood vessel formation, angiogenesis, and in case of cancer, it would be invasion or inflammation, immunosuppression, genetic stability, etc. So it means to show that MIR21 is a very important, it can be considered as an important pathological active microRNA. So what I did was, first, because I was working with diabetic wound healing and we were wanting to see about we are studying the cell migration studies. So using fibroblast cells, treated the cells with a precursor form of synthetic MIR21 or oligonucleotide sequence of MIR21 to sponge, which will act as an anti-MIR21. So we created a wound on the we scratched the cell surface to create a wound, and then we added either the MIR21 mimic or the MIR21 antagonist. And after 18 hours, we studied to see whether the cells are migrating into the wound area. So you find here that in the MIR21 treated cells, we have more amount of cells migrating into the wound area in comparison to the control. And when MIR21 was suppressed by the antagomer, we have less amount of cells migrating into the wound area. So it meant to say that MIR21 was necessary for cell migration. So obviously here, as we say, it is like invasion. We can match like it is important for cell migration. So this is the video of the cell migration studies that we do. These are fibroblast cells. We grow the cells as a single layer, and we scratch the center of the plate to create a wound, and then we monitor the cells to watch their migration over a period of time. Okay, so then I studied, as a molecular biologist, of course, I'll be interested in studying the mechanism of what happens. So I found out that the MIR21 functions through two important molecules, TGF, transforming growth factor beta 1, and nuclear factor kappa B. So what we found out is that using Western blot analysis, and this is the immunofluorescence studies, this is EMSA studies, and this is chromatin chip immunoprecipitation assays. Using different assays, we found that TGF beta was necessary to activating NF kappa B activation, NF kappa B pathway, and the NF kappa B P65. I'll explain about this more later. NF kappa B P65 interacts with the MIR21 promoter and increases its expression. So that was about wound healing. Next was my MIR21 and inflammation. The focus was on inflammation. So MIR21 is associated with inflammatory activation. It's upregulated in many of the pathologies that are associated with inflammation, but its precise role is not very clear. So macrophage polarization, the polarization means macrophage, they can either be pro-inflammatory or anti-inflammatory depending upon the situation. They're not permanently fixed. M1 is the pro-inflammatory macrophage phenotype. M2 is the anti-inflammatory phenotype. M1 can become M2. M2 can become M1. It's not like it's a transient phenotype. Depending upon the situations, they can change. So the resting macrophages are called as M0. So to M0 macrophages, we added MIR21. And then using flow cytometry, we studied the population of M1's population and M2 anti-inflammatory population using specific markers. So the cells were stained with the specific markers and subjected to flow cytometry. And we see that in the MIR21 treated cells, there was a shift from 36% to 20% shift of the M0 macrophages into M1 pro-inflammatory macrophages. So this showed that MIR21 was responsible for the pro-inflammatory, was inducing pro-inflammatory status of macrophages. And as in the morning, Dr. Beta CV already explained about NLRP3 inflammasome. NLRP3 inflammasome is important. One of the important complexes involved in inflammation. So when we studied the cells treated with MIR21 or cells treated with MIR21 and anti-MIR21, or cells treated with MIR21 with NFI. NFI is NF kappa B inhibitor. So we find that in MIR21 treated cells, the NLRP3 ASE caspase is increased, expression is increased it is nullified, reduced when we have when the MIR21 is sponged or sucked up, used up by the anti-MIR21, so that MIR21 cannot bind to the other proteins or mRNAs. And same was found in the case of NF-kappa B inhibitor. If we inhibit NF-kappa B, MIR21 does not have a functional effect. And the NLRP3 complex will cause the activation of caspase-1 activity, which is the most ultimately leading to IL-1 beta and IL-18 release. So again, this was explained by Dr. Beta Sivi, so I will not go into the details. And this is the immunofluorescence. The blue shows the nucleus dyed with the host 33342, and the red dots are the active caspase sites. So when MIR21 is added to the cells, we have more amount of active caspase 1. Anti-MIR21 and NF-kappa B inhibition reduce the amount of caspase 1 activity. So these results show that MIR21 requires NF-kappa B for its inflammatory activity. The next focus was exosomes. So what are exosomes? Exosomes are extracellular vesicles. There are one kind of extracellular vesicles, like they are derived from within the cells. So in the cytosol, we have the endosomes. The endosomes form multivesicular bodies, and they fuse with the membrane, cell membrane, and then they are released out as exosomes. So these exosomes are specific. They have specific markers in their on their cell surface, and what do they have inside? They contain a load of different molecules like mRNA, DNA, miRNA, proteins, and also growth. Of course, growth factors are also proteins. So this is the cargo. We call this as the cargo of exosomes. So the exosomal cargo is not the same with between all the cells. It depends on it varies with the cells. And what happens when the exosomes are released from the cells? They are ready. They can be easily accepted by recipient cells. So depending on the conditions, if favorable conditions, the exosomes, the surface markers, surface proteins will bind to the recipient cells, and once they bind, they can get fused with the cell membrane, and they get into the cell's recipient cell, and they can have their functional effect in the recipient cells. So exosomes are like the courier boys. They can transfer the machinery, the cellular machinery from one cell to another cell. And these exosomes are found in our blood, urine, tears, breast milk, and even the sweat also. So almost all of the biological fluids, saliva also, all of the biological fluids are rich in exosomes. So exosomes now are considered as are being tried for to prepare like biomarkers for cancer or for Alzheimer's disease. For various diseases, they are being tested so the exosomal cargo is being studied for the biomarker potential and even for target therapeutic potential. So like, for example, we find that these exosomes are found to have effect in almost all of the physiological functions. And that's why they are important study materials in the research area. So with this in mind, we collected the exosomes from cells, normal cells, as well as from MIR21 treated cells, macrophage cells. And this is the electron microscopic. These rounded materials are not cells. They are the exosomes secreted by the cells. This is the electron microscopic view of the exosomes. And studied the structure, the NTA-based size analysis showed that the exosomes varied. They are small, like about 35 to 40 nanometer in size. And we also profiled the exosomes to show that, to find out if they are either actual exosomes or like apoptotic cell bodies or some other extracellular vesicles, using specific markers for the exosomes. So CD63, CD9, flotillin 1 are markers of exosomes, and we found they are expressed in these bodies. So that means it proves that they are exosomes. In addition to that, we also studied whether the exosomal cargo contained MIR21, because here we should see that we have nucleic acid. So whether MIR21 is expressed in these exosomes, we did real-time PCR and we found that in those MIR21 was present in the exosomes in both control and exosomes secreted by the control cells as well as the MIR21 treated cells. But in the cells that were treated with MIR21, the amount of MIR21 in the exosome was very high. So that means that the cells produced MIR21 and that was also secreted into the exosomes in large quantities. So that means, again, what it means is that this MIR21 inside the exosome, when it goes into a recipient cell, the amount of MIR21 in the recipient cell should also basically increase. For that, we first have to check whether these exosomes can be taken up by the recipient cells. We used a red colored dye, PKH26. We stained the exosomes with this dye and we added it to the macrophage cells, normal resting macrophage cells. And we find that the blue color is the nucleus, and we find that the red color dye has gone into the cells. So that means successful uptake of the exosomes by the recipient cells. And as I said, if the cells take up the exosomes, does it affect the amount of MIR21? Yes, it does affect. We did real-time PCR of the MIR21 of the recipient cells, and we found that in comparison to the exocells that were treated with control exosomes, the cells that were treated with MIR21 rich exosomes had a high amount of MIR21. And when we treated these cells with the MIR21 inhibitor, the amount of MIR21 was decreased. And this PDCD4 is a target mRNA of MIR21. So if MIR21 increases, PDCD4 basically should, if it is active, if the MIR21 is active, the PDCD4 amount will reduce either at the RNA or at the protein level. So we found that even in the RNA level, the PDCD4 was decreased. So it means that the MIR21 that was transferred through the exosome was functionally active in the recipient cells. What about the inflammatory profile of the recipient macrophages? So we have the cells that received the control exosome, cells that received the MIR21 rich exosomes, cells that received the MIR21 exosomes along with the anti-MIR21. So we find that this set of pro-inflammatory cytokines, pro-inflammatory proteins, increased in cells that were treated with MIR21 exosome. And the anti-inflammatory IL-4 and IL-10 interleukins decreased. So this basically shows that the MIR21 that is transferred through the exosome is very functionally active and it causes a pro-inflammatory profile of the recipient macrophages. So this shows the importance of MIR21 in different pathological conditions or pathological events that lead to diseases. So and that's why our interest in studying more about this MIR21 as a therapeutic target was induced. So here I'll talk about the NF-kappa B. So what happens is when we have a stimulus, inflammatory stimulus enters into the cell, we have a set of NF-kappa B family called as IKK complex. We have P65 or REL-A, P50, and usually REL-A, P50. This forms are always in a complex with IKB alpha. IKB alpha is an inhibitory subunit of NF-kappa B family. So when IKB alpha is binding with this complex P65, P50 complex, these proteins cannot be activated. They are in dormant state. But when an inflammatory stimulus takes place, enters into the cell, the signal causes the phosphorylation of the IKK complex. So when IKK is phosphorylated, which means it's activated, it also phosphorylates IKB alpha and these proteins. IKB alpha, when it gets phosphorylated, it gets released from this complex and it gets ubiquitinated and it's degraded. On the other hand, these two proteins, P65 and P50, when they get phosphorylated, we call them as activated forms. They get into the nucleus. From the cytoplasm, they get into the nucleus. And in the nucleus, they bind to target DNA. For example, MIR21 has a binding site for P65. So the P65 will bind to the MIR21 promoter and it will increase the MIR21 expression. And this further will increase the expression of other proteins. So NF-kappa B is a very popular target for therapeutics, because we can target NF-kappa B by inhibiting IKK phosphorylation, or by preventing the ubiquitination, or by preventing the nuclear translocation of the P65, P50 complex. Or even if it enters into the nucleus, we can prevent the DNA binding activity of the complex. So there are so many ways we can prevent the NF-kappa B activity. So my go-to strategy for therapeutics is following the father of medicine, Western medicine, Hippocrates. He said, our food should be our medicine and our medicine should be our food. It's the same as what we have in Ayurveda. So food is our medicine. So we look into the food and this time we are focusing on retinoic acid. Again, retinoic acid, Dr. Beta Sivi had explained about it. So I will not go into the details. So it's a active metabolite of vitamin A, and it has various important roles in various biological processes. Basically, it starts from the beta carotene, carrot. It's hydrolyzed into retinoic acid, retinol. Retinol is converted into retinal. Retinal is converted into retinoic acid. And this retinoic acid is the active metabolite of vitamin A. And this retinoic acid is already known to have large, major, important roles in anti-inflammatory, management of many inflammatory diseases. So what we did was, using macrophage cells, we induced inflammation by adding LPS, lipopolysaccharide. So we studied the expression of the different inflammatory, anti-inflammatory and pro-inflammatory genes. So you find this, the first column is the control cells. The second one is the cells that were treated with LPS. So you find that pro-inflammatory genes, IL-1 beta, TNF, INOS, IL-6, they increased in LPS-induced inflammation. But when we treated the same cells, co-treated with retinoic acid, the LPS, inflammatory effect of LPS was reduced. So that means RA is retinoic acid. It is functional in inhibiting the pro-inflammatory cytokine production in macrophage cells. And we did actually, this work was done by one of our PhD students, Dr. Queen Nurama. She graduated recently from her PhD course. So this was her PhD data, and which she did a lot of for some other studies. I'm not showing you the details, but in summary, she found that LPS can induce IKK alpha B phosphorylation. IKK, as I explained earlier, IKK alpha phosphorylation can cause the P65 to go into the nucleus. It can bind to the MIR21 promoter. It increases the expression of mature MIR21, and it causes the increase of inflammatory cytokines, and the inflammation increases. When retinoic acid is there, IKK alpha is phosphorylation is prevented, and because of this prevention, all the other downstream activities are prevented, and inflammation is also prevented. So this is the therapeutic route of retinoic acid in inflammation, LPS-induced inflammation. We are now studying the nanotherapeutics. So the retinoic acid, we conjugated them with zinc nanoparticles to make retinoic acid zinc nanoparticles, and studied the therapeutic. We are studying the therapeutic potential. So cells, this is the cells treated with LPS, and this is the cells treated with LPS and the retinoic acid nanoparticles. So if we can see, there are some green. The blue is the DNA nucleus, and there are some green dot. There are many green dots in this LPS treated group, showing the ROS generation. But when we add retinoic acid, the dots are minimized. So show retinoic nanoparticles are previously for inflammation. So the caspase 1 activity. So we have the LPS treated cells. We have red colored areas that show the active caspase, and in retinoic acid nanoparticle treated cells, the amount of red color is reduced. So we are still working on this, so I'm not able to show you further experiments. Hopefully in the near future, we can share our research data about this nanoparticles. So far, I've just explained to given you a brief outlook of my research road, that's MIR21 induced inflammation, working on nanoparticles, exosomes, and therapeutics. But so what? If now we know that MIR21 is causing inflammation, we know that it is working through exosomes, creating a paracrine effect, and we know that retinoic acid can inhibit this induced inflammation. So can we use retinoic acid as a drug? We doubt it, because as everyone must be knowing, this is the problem with the current medical system is we have one single drug for everyone. So, but gradually people are realizing that one size does not fit all. We know that same patients suffering from the same type of cancer, some of them get cured completely, but some of them cannot get cured. So although it's the same drug and the same mechanism should be acting within our bodies, it's not the same. So it's like one size will not fit all. We need personalized medicine, just like Dr. Prasad had explained earlier. So this is the physician way long back, more than a century. He had said, no two individuals of a species are absolutely identical in bodily structure. Neither are their chemical processes carried out on exactly the same lines. So this he had already told even before we have the current biomedical system. So that's the reason why the same disease can some of them can get cured with the same with one drug, but the others cannot get cured with the same drug. So the road forward would be to have individualized treatments, that is based on each personality or personal genomics of the individuals. we need to have different therapies. So this is currently in process, but it's very time consuming and consuming also. It's costly and it's time consuming. So we need to have something that is people friendly. So the road forward for personalized medicine would be a mix of pharma. Again, the same concept was talked by Dr. Prasad. So we have pharmacogenomics. This is what we are currently doing now, studying the effect of different pharmaceutical products on the cell biology, molecular biology. And we also have to study the personal genomics. Individual genomics, the genetic modeling have to be studied for each individual, and based upon these two data, we need to prepare medicines for each person. So the route one would be, again, this Dr. Prasad also mentioned about this Ayurveda. We have the Vata, Pitta, Kapha. So it is not the overall, it doesn't give an overall view of the individual personality. It just classifies people based upon their personality. But combining that with the gene sequencing, we need to add this data, the traditional knowledge with the current knowledge we need, and to combine it with the pharmacogenetics is one route. The second route is thinking out of the box, like Einstein had said. Future medicine will be the medicine of frequencies. So this is where we come. This is one example of frequencies. So this is actually, we can call it as the song or a dance of the cells. If our cells could sing and dance, this is how it would be. This is the song of a healthy cell, and this is supposed to be the song of a cancerous cell. So this technology term was coined by Dr. James in 2002, and he coined a term called a sonocytology. So what he did was he found that our cells, every cell of ours, are vibrating at a frequency. So he developed an instrument, a microscope called as thymoscope, and to pick up the frequencies of the cells. The cells are emit a frequency because some motor proteins, when they are functioning on the cytoskeleton, they like walk on the cytoskeleton, and they create some vibration of the cells. And it picked up these frequencies, and when it passed on to a surface of water, and when light is provided on the surface of the water, I believe this is what this is the pattern we can get. So the healthy cell and the cancer cell. So this is a completely different pattern. So okay, so this is just a video to see that. I hope you can hear the song. Can you hear the song of the cells? So this is a healthy cell. Can you hear the sound?
No, ma'am, it's not audible.
Yeah, even I'm not able to hear it. Anyway, it's on the YouTube, so if you can just hear the sound, it's more interesting. And this is a stressed cell. This is the sound of the stressed cell. So this is just an example of out-of-the-box thinking, mixing it with the past, that traditional knowledge that is brought about, that has been given to us by our ancestors, our predecessors. So the road forward, in my opinion, the best route would be to convert the cancer cells into healthy cells and using the food. So this is our predecessors, and this is our current state. And the future road would be to use these two to get a balanced health for everyone. So with this, because like it right now it seems like out of the box, and it's like no, this is not impossible. But 20 years ago, none of us dreamt of anything called as Wi-Fi or Bluetooth or even smartphones. They were not existing, but now they have become so mandatory. They are like, without them we cannot, our daily life is like handicapped without them. So similarly, you youngsters, you have the, like, now you're standing on the verge of you're standing on a new horizon, and you have the benefits of our traditional knowledge as well as the present technology, advanced technologies. So I would request our youngsters, whoever is interested in research, to take up these basic medical research. Like, these are the stepping stones for clinical research. So please use these technical advancements. And since you're in the biotechnology engineering part of it, it's easy. It's more like you're more at an advantage of mixing physics and biology together for the benefit of mankind. And please come up with some innovative, simple, cost-effective, and user-friendly therapies for the benefit of mankind. So with this, I would like to end my talk. So first, I would like to give my acknowledgements to, of course, the University of Miyazaki Faculty of Medicine, and also the Faculty of Engineering for giving us, always having, cooperating with us, and the Japan Society for Promotion of Science. It's like, without this project funding from the government, we will not be able to do our projects. So we have been lucky, we have been guided by their project fundings. And then importantly, thanks to Japan Science, JST, Japan Science and Technology Agency, which annually organizes calls forth for this Sakura Science Exchange program, which we can use. We are just utilizing the opportunity given to us by the Japanese government. So immense gratitude to the Japanese government. And last but not the least, I would like to thank N.M.A.M Institute of Technology, Nitte, especially Dr. Kamath, for being very cooperative and understanding in organizing today's event. Thank you, everyone.
Thank you, ma'am, for your wonderful insights on the role of microRNA in diabetic wound healing, MIR21 as an important pathological microRNA, and NF-kappa B targeted therapeutics. The session is now open for discussion. If anyone in the audience has questions, you may ask now.
We have a question. In case we are interested to intern at your department, what is the opportunity?
You are in your bachelor's, right? So
Yes, ma'am.
okay. So in Japan, if you want to become like if you want to do research, become a researcher, we need 18 years of experience, educational experience. So because you have 4 years, you will be when you complete, you have 4 years of bachelor's. So 12 years of schooling plus 4 years, 16 years you will have. So you need to do 2 more years of master's, and then you can come here for your research. But there are some universities like VIT. We have this 6-month semester abroad program. So where master students can come here, and in our lab they can, in our university they can do internship. So if Nitte University, I think we are trying for a SAP program with Nitte University also. So if that is successful, then during your masters you can come. You'd be able to come here for doing your internship. Did I answer your question?
Yes, ma'am. Thank you so much. We have one more question, ma'am. How are exosomes useful in treating Alzheimer's?
Not only Alzheimer's. I told you like they are now being tested as biomarkers. So potent like early diagnosis exosomes, like just a drop of the blood contains a lot, not even blood, a drop of urine or even teardrops or saliva contain exosomes. So those exosomes, like there are some markers specific for Alzheimer's, and if those markers of Alzheimer's are detected in exosomes, then they can be used as a diagnostic, like biomarkers. So early stage of Alzheimer's can be detected by exosome. So it's not only what do you call it? It's not restricted only to Alzheimer's, even for cancer, early stages of different cancers. All cancers are not the same. So different, like again, Dr. Prasad had earlier mentioned, different cancers have different markers. So based upon the early, these exosomes, we can, without biopsy, like non-invasive method, this is a non-invasive method, we can use them as biomarkers.
Thank you once again for your enlightening presentation, ma'am.
You're welcome.
The Sakura Science Online Exchange Program not only aims at educating young individuals, but also aims to provide a platform for students in the science field to present their innovative ideas. We now have a few groups of students who will present their ideas through flash presentations. I would now like to welcome the jury members for the Ideathon. Dr. Ujjwal P., Professor and Head, Department of Biotechnology Engineering, NMAMIT Nitte. Dr. Kaoru Ohe, Department of Applied Chemistry, University of Miyazaki, Japan. Dr. Shamaprasad Sajankela, Associate Professor, Department of Biotechnology Engineering, NMAMIT Nitte. Dr. Radha Madhyastha, Assistant Professor at the Department of Cardiovascular Physiology, Faculty of Medicine, University of Miyazaki, Japan. Dr. Harish Kumar Madhyastha, Associate Professor, Department of Cardiovascular Physiology, Faculty of Medicine, University of Miyazaki, Japan. A warm welcome to all the jury members. Jury, please note, first up is a presentation on neovascularization using tissue engineering by final year BE Biotechnology students, Ms. Anjali Rao Kalbavi and Ms. Pratiksha B. You may now begin with your presentation.
I hope I'm audible and the screen is visible.
Yes. Yes.
Thank you. Good afternoon, everyone. Today I'm going to talk about neovascularization using tissue engineering. In a seminal paper in 1993, Langer and Vacanti defined tissue engineering as an interdisciplinary field that applies the principles of engineering and life science towards the development of a biological substitute that restore, maintain, or improve tissue function. The three original pillars of tissue engineering as proposed by Langer and Vacanti are matrices, which include natural and synthetic scaffold constituting extracellular environment of a particular tissue or organ, isolated cells such as stem cells or differentiated cells to restore the function of a diseased or injured tissue, and tissue-inducing substances, which is an array of biomolecular technologies capable of controlling cell behavior. Over the past centuries, there has been an increasing incidence of ischemic diseases such as atherosclerosis, arterial disorders, due to the drastic change in the diet of lifestyle and people, and is the leading cause of mortality and morbidity. These diseases have resulted in ever-persistent demand for vascular conduits to reconstruct or bypass vascular occlusions and aneurysms. Although synthetic grafts are available for replacing the occluded arteries, surgical complications associated with harvesting vessels, frequent shortage of allogenic grafts, and immunologic rejection of large animal-derived vessels, which can be fatal, are the reason why tissue engineering is being looked into as a cheap, effective, and durable alternative to the existing methods. Here we are going to talk about the construction of two types of blood vessels: large vessels less than 6 millimeter in diameter and smaller vessels which is less than 1 millimeter in diameter. Construction of arterial blood vessels. So creating a tubular structure with biomaterials involves three commonly used methods. First is sheet rolling, where a sheet of desired biomaterials with or without cells is prepared and rolled over a mandrel into a tubular structure. Matrix molding, where the shape materials are injected and cross-linked inside annular molds so as to obtain tubular scaffold. And direct scaffolding, where the polymers are directly formed into a tubular scaffold by physical carving or electrospinning on the surface of a rotating cylinder. Grafts can also be sourced from decellularized xenogenic tissue, often pig. The next step is cell seeding, that is inserting of cells onto the prepared scaffold. Some of the methods used is static seeding techniques, dynamic seeding techniques, and so on. Once the graft is fabricated, a tissue-engineered blood vessel may not yet possess sufficient strength to support the physiologic blood pressure and requires further maturation in a bioreactor. And the strategies used to induce this adaptation are utilizing a pulsatile pump to induce flow through the inner wall of the graft, and application of external stimuli such as shear stress and matrix stretching. So even with the technical advances, most of the current bioengineering techniques are unable to create a patent perfusion vessel. This critical challenge can be overcome by exploiting cross-kingdom contribution within the same bioengineering platform. Plant cell walls is composed of cellulose, hemicellulose, and pectin, and these compounds are known to be compatible and promote wound healing. In recent times, decellularization techniques are being applied to different plant species and tissues in order to generate acellular pre-vascularized tissue-engineered scaffold. So the idea that I would like to propose here is utilizing bamboo stem and associated xylem vessels for the construction of arterial blood vessels. The bamboo stem of desired length and thickness is excised and decellularized so as to obtain a scaffold. The cells can then be seeded on the inner wall of the scaffold, and as the cells proliferate, it takes the shape of a tube. There is also a possibility that the cells penetrate into the spaces of a scaffold, surround the xylem vessels, thereby creating a vasorum-like structure. Thus, the vascular conduit so obtained is very similar to the one present in the human body. The next part will be explained by Pratiksha.
Construction of microvessels. Microvessel structure that lacks the well-organized elastic lamellae of arteries and the valves of veins. Diameter of the microvessel is less than 100 micrometer, depend on the material to be exchanged. Microvessels are made up of initial capillaries and small collecting lymphatics. Primary function of microvessel is to exchange fluid, solutes, and cells between the lumen and surrounding tissues. Biomaterials used. Biomaterials used for the construction of microvessels are hydrogel, collagen, fibrin, which provide a suitable environment for angiogenesis. Endothelial cells, human umbilical vein endothelial cells, induced pluripotent stem cell derived endothelial cells are most commonly used for the fabrication of microvessels. Engineering of microvessels. Microvascular fabrication can be divided into two strategies: top-down, bottom-up. Under top-down approach, the resulting geometry and architecture of the vascular bed is pre-designed before cells are introduced. 3D printing technique, where sacrificial materials are used in order to generate filamentous network structure, which is then dissolved to create a microfluidic channel, where the endothelial cells are seeded later. Laser degradation. Biomaterials with or without cells cross-linked with hydrogel. Laser is used to scan the pre-designed geometry to degrade the material. Layer by layer assembly. The pre-designed 3D structure is fabricated additively, where each layer is fabricated separately through lithography. All the components are aligned and fixed together mechanically or chemically through ultraviolet linking. Under bottom-up procedure, angiogenesis-driven vascularization. The endothelial cells seated on the walls of parental microfluidic channel, chemical and physical stimulants are provided where the vascular sprouts are observed. After days and weeks, the scaffold is formed, later implanted. Vasculogenesis. Self-assembly of endothelial cells into multicellular network forming vacuoles that fuse to form a vascular lumen by providing physical and chemical stimulants. After days and weeks, network of vacuoles formed which is then implanted. The future idea. We are planning to use hemicellulose from cannabis sativa as bio-ink, where hemp contains 32.78% of hemicellulose. Antisense technology can be used in order to overexpress the hemicellulose gene to produce more content of hemicellulose. Conclusion. Millions of animals are estimated to be used in research, which has led to the increased energy consumption and waste generation. And also the plethora of ethical concerns are involved using animals for generation of blood vessels. So plant tissue culture and engineering can be used as a green solution for number of problems and application of regeneration medicine. These are the references. Thank you.
The session is now open for discussion.
Organizer, shall we have to ask any question or what?
Yes, sir.
If you have any queries, then
Okay. I have a small, hypothetical question.
Yes.
Excellent presentation, nicely coined up, and importantly, you maintain the time. That is most important in the rapid presentation.
Thank you, sir.
You are using xylem as a bio-inspiration model, or a xylem itself you wanted to use?
What I was trying to tell is using the bamboo stems, like using plant tissue culture technology, we can synthesize bamboo stems of various thickness, diameters. So we can see the cells on the inner surface of the bamboo. And so, like, if the bamboo of desired length is taken, then the cells will, like trace the scaffold, no sir, like the inner surface of the scaffold.
Good.
So what happens is that you already get a tube-like structure. The bamboo itself is in a form of a blood vessel, tubular blood vessel.
True.
And when the cells trace this scaffold, it's you get a blood vessel, and when they penetrate into the spaces, you get smaller capillaries.
Yes.
So if you this is very similar to the one that you get in the body, because it's not just one blood vessel to any organ. It's a main blood vessel which is interconnected by several smaller capillaries. So this using bamboo scaffold, I feel, will make tissue engineering of blood vessels very simpler to the ones that we have right now.
Good idea.
Thank you, sir.
OA 先生、何がquestionありますか?
So, I have one question. So, next presentation, second presentation, future ideas.
Yes, ma'am.
So, why will you select the hemp hearts?
Yes. You see, ma'am, if we wanted to modify the hemicellulose content, so the other crops that have high hemicellulose contents were sugarcane and cotton crops, and getting an ethical clearance to genetically modify such edible crops is quite difficult. But hemp already has quite a high content of hemicellulose, and it's only being used as for extraction of anesthetic products. So we can easily get ethical clearance and modify the plant to increase the hemicellulose content, and we can also extract it quite easily.
So, first presenter chose a bamboo.
Yes.
I don't know. So, the advantage of bamboo and hemp.
I didn't get you, ma'am.
So hemicellulose is very important, the material.
Yes, ma'am.
So, and how about bamboo hemicellulose content?
Yes, ma'am. That can also be studied, but we primarily took hemp because of ease for extraction and modification. We decided on hemp for hemicellulose content and bamboo for direct preparation of scaffold, so that we get two different ideas. This can also be tried, ma'am, no doubt. But that's it.
Okay, thank you very much.
Thank you, ma'am.
Very nice idea.
Thank you.
Can I ask a question?
Sure. No.
So you are planning, like your idea is to use bamboo for larger vessels and then use hemp for 3D printing of micro vessels, right?
Yes, ma'am.
Why can't you use 3D printing for the larger vessels?
It is possible, ma'am, but it's quite expensive. That is the reason why we are taking bamboo, because it's naturally available. And
it's naturally okay. So how about its biodegradability? Like how fast it degrades, and you're using it only as a scaffold, right? You're not going to, like, after that you're going to remove the cells collect the cells from the scaffold, or are you going to planning to use the bamboo scaffold as such?
It depends, ma'am. If the endothelial cells are strong enough to hold themselves on their own, then we can degrade the bamboo scaffold using cellulase enzymes. So that will help in degradation of the cellulose and hemicellulose content, and we'll get only the vessel.
Okay. Interesting.
Thank
nice
you.
Presentation. Thank you.
Thank you, ma'am.
Next up is yes, ma'am. Next is a presentation on sonochemical proteinaceous microspheres for wound healing by Ms. Rashmita V. Shetty and Ms. Anushri B., Final Year B, Biotechnology. You may now begin with your presentation.
I hope screen is visible and I am audible too.
Yes, Rashmita, continue.
Okay. Good afternoon, everyone. We are talking about sonochemical proteinaceous microspheres for wound healing. These are our contents. Wound is the result of disruption of normal anatomic structure and function. Based on the nature of repair process, it can be classified into acute and chronic wounds. Acute wounds are tissue injury that heals completely with minimal scars within expected time intervals, maybe 8 to 12 weeks. Chronic wounds arise from tissue injuries that heal slowly, not beyond 12 weeks, and it often reoccurs. Acute wounds have low levels of protein degrading enzymes, whereas non-healing chronic wounds contain elevated levels of proteases. Sonochemistry is the use of ultrasound to enhance or alter the chemical reactions. Microspheres are small spherical particles with diameters in micrometer range. Generally, they will not have any fluid inside as opposed to microcapsules. And this is the formula for the microspheres formation. Here, protein I and protein F are initial and final protein concentration respectively. Zeta potential and size distribution of microspheres were determined by electrophoretic laser Doppler anemometry and photon correlation spectroscopy. Coming to methodology, determination of PP activity laws over time. Activity of PP was measured by adding enzyme with reaction buffer at pH 8. Reaction was started using synthetic substrates, and cleavage of these substrates were monitored spectrometrically. One unit is defined as amount of enzyme that will hydrolyze synthetic substrate per minute. Inhibitory activity of organic solvents was examined by adding proteinaceous microspheres to a fixed amount of PP solution. Incubation was carried out at determined time points. Aliquots were collected to monitor the decrease in the elastase activity. Measurements were recorded and expressed as mean value plus or minus standard deviation. Cytotoxic screening. Proteinaceous microspheres were tested for cytotoxicity. BJ5TA cell lines on normal human skin fibroblasts was maintained. Cells were maintained in humidified atmosphere and culture medium was refreshed every 2 to 3 days. Cells were seeded in tissue culture polystyrene plates and exposed to different microspheres concentration. Cell viability was determined, and quantity of resoreuffin formed is directly proportional to the number of viable cells. The two-way ANOVA was employed with statistical significance. Now I request Rashmita to take over.
Coming to the part of results and discussion, characterization of proteinaceous microspheres. The different proteins were focused in the study of microsphere formation yield, particle size, polydispersity index, zeta potential, and morphology. All experiments were done with 3 different organic solvents, while N-dodecane presents 12 carbon-linked bisaturated bonds, whereas vegetable and mineral oil has different degrees of saturated and unsaturated fats. At first, to determine the successful yield of protein, the Lowry procedure was used. The proteinaceous microsphere has high yield on microsphere formation independently on organic solvents used. However, this exhibit a slight difference of mean size when vegetable and mineral oil were applied. The linear zigzag organization of single bond enables the chain to be lined up close to each other, and intermolecular interactions like van der Waals force and all, which promotes size reduction and explains the results obtained for N-dodecane. The presence of double bond produces kinks in the geometry of the molecule. The silk fibroin presents the highest Z average values when compared to bovine serum albumin and human serum albumin. There was no difference on the yield of microsphere formation particle size when the proteinaceous microsphere were prepared in PBS medium or in water. Another characteristics of polymeric microsphere, that is of extreme interest, is zeta potential, which is a function of surface charge of potential a particle. The results show that zeta potential value drops significantly when protein microspheres are prepared with PBS solution. Suspension stability is another parameter for material characterization. The stability studies were performed over 4 months, analyzing the macroscopic aspect and measuring the size distribution and zeta potential. Coming to the part of inhibitory effect. The inhibitory activity. The major goal of this work is the development of proteinaceous devices with a specific biological functionality. That is basically the inhibition of elastase in chronic wounds. It is important to determine the inhibitory activity of the different organic solvents incorporated into protein devices. The highest concentration of proteinase microspheres used were incubated with PPE. It was achieved that more than 40% of the activity loss for all the proteinaceous microsphere prepared with different solvents after 5 minutes of incubation. Both mineral and vegetable oil demonstrated a higher activity loss, especially when silk fibrin used as a carrier. Coming to the figure that is being depicted, it says that BJ5TA cell viability at 24, 48, and 72 hour of the culture with different material solutions and different concentration of human serum albumin, and silk fibrinonin microspheres incorporating vegetable oil. Statistically significant differences are observed. Coming to the part of conclusion, microemulsion system was characterized and selected. Microemulsion were explored for their ability as potential elastase inhibitors. The effective nature of protein should be considered. Once that silk fibrin demonstration to be more capable of transferring vegetable oil in aqueous solution under condition of pH properties, which is mimicking the wound fluid. The cytotoxicity screening indicates that the developed protein particles are not cytotoxic in wide range of concentration. Coming to the future ideas, the wound healing is rather complex. Although the understanding of these properties may help to support the further refinement of protein devices for improved wound healing. In this research, human neutrophil elastase is used, which is basically a protease enzyme that when expressed, it can cause emphysema. Emphysema is nothing but a lung condition that causes shortness of breath. Hence, other protease enzymes like bromelain that can be used, which plays a dual function of removing the scars as well as it cures the deep burns with very less side effects. These are the references. Thank you.
The session is now open for discussion. If there are no queries, can I move on to the next presentation?
Yes, please go ahead.
1 minute. Sorry to
Yes,
interrupt. I was not clear whether this is Miss Rashmita and Miss Anushri's original data, or is it someone's work that they have expressed here?
Ma'am, the future ideas is our idea, but this is a research topic which is ongoing.
Ongoing in your university, or?
No, it was published in one of the textbook, and we have taken points from there.
Oh, okay, fine. So the future objectives are yours, right?
Yes, ma'am. Yes.
Okay, fine. I got it. Thank you.
Thank you.
Are there any more questions?
So I have a question. So you explained that silk protein is I don't understand. Silk protein particle is good. So you what did you say? Explain the protein.
Hello.
Any kinds of protein except silk protein.
Ma'am, that is what I explained. They have made use of bovine serum albumin, human serum albumin, and silk fibrin. Out of this, they found that silk fibrin, it has the highest Z average values when compared to BSA and human serum albumin. Hence, they have made use of silk fibrin for the further studies. And coming to your next question, according to our future aspect, it is we have mentioned about bromelain enzyme. So even that can also be used because it has very less side effects when compared to that of human neutrophil elastase.
Mhm. So you check the zeta potential of a protein particle. So positive charge is good or not. So I don't know. I'm stranger in your. I don't know your research field. So I want to know the zeta potential, positive or negative, is which is good.
I'm sorry, we did not get you.
You explain the zeta potential of protein particle.
Yes, ma'am.
Yeah. So, and I want to know the zeta potential is negative or positive. Your material for material, which is good, which is best, negative or positive?
Ma'am, basically it should be negative. It is better
Negative.
if it is negative.
I want to know the reason. So target is negative, positive, and so and particle zeta potential of the best of the particle surface zeta potential is negative. Is it true?
Ma'am, the negative symbol is like the net charge scattering object. If there is more scattering, then it is negative. So we need the substance to be more scattered. Hence, it has to be negative.
Thank you very much.
Thank you, ma'am.
Jury, please note, next up is a presentation on zebrafish knockout model for scientific research by final year B biotechnology students, Ms. Neha B. Salyan and Mr. Mohammad Atif G. You may now begin with your presentation.
Yeah. Am I audible and my screen is visible?
Yes, go ahead.
So good evening, everyone. I and Atif is going to present our idea on zebrafish knockout model for scientific research. So zebrafish is an important biomedical model because zebrafish embryos are transparent and they develop outside the uterus, and this unique development process allows the scientist to study the details of the development starting from the fertilization and continuing to the development through zebrafish and human. Though zebrafish and humans have difference in basic nutrients requirements, different metabolic mechanisms may not be needed to keep the balance between the production and the utilization of the energy. Several organs are involved, including brain, intestine, liver, skeletal muscles, and adipose tissues. So this is a classification. Zebrafish is small, about 3 to 4 centimeter long. They are freshwater fish belonging to Cyprinidae family, and it is originated from East India, which has mini tank size of 10 millimeter gallon. They are omnivorous and breeding egg layer. They are easy to care and maintained in a pH of 6.5 to 7. And
The gender differences are the both sexes have two pairs of marbles and same stripes, but females are usually larger. Males are a bit smaller and more slender than females. Male zebras appears more torpedo shaped, while females tends to have large belly. Movements of a male will be faster in the water than female. Graphene is expensive to purchase in a large quantities, which has led to an interest in graphene oxide, which is less expensive alternative for graphene. Graphene oxide are particularly interested in medical field due to their capacity for modification throughout their production, which has diverse medical application. So, graphene oxide loaded with the chemotherapy drug doxorubicin, which have been used to deliver the drug directly to the cancer cells in vitro. Graphene oxide are ideal pH sensors for cancer detection due to their sensitivity to pH changes, and zebrafish have become well established model in toxicological studies due to their rapid development, optical transparency throughout embryonical and larva development, and molecular homogeneity to humans with added benefits of exploiting a whole organisms rather than single tissue or cell type. So in this diagram, 50 microgram of per ml MGO will be exposed to AG and UP water control. MGOs will be imaged for 1 hour after initial exposure at 6 hour post exposure. These images were taken from BZX series. Scanning electron microbes will be identified deep cracks in the yolk region of 6 HPF embryos, which is exposed to 50 microgram per ml MGO, where embryos in a UP water possess two superficial cracks. These deep cracks are identified occurred in the yolk cells. So, genetic engineering in cancer research. Zebrafish has gained a robust model for cancer research because the large number of progenitors can be obtained and they are cost effective in maintenance. Dynamic visualization of tumor growth in vivo. So the transparent embryos, after 3 months of a rapid generation times, they form an adult. From this, they are very small and easy for the maintenance. And these fishes can be used for carcinogenesis models. And the progenitors which are obtained will be 100 to 200. They're used in a large-scale genetics and chemical screens. And they're also used for transgenic genes knockdowns models. And one such zebrafish is used in a pediatric cancer. Pediatric cancer is one of the leading cause of death in children and adolescents, and it is mainly comprised of leukemia, cancers for nervous systems, and sarcomas. The use of this zebrafish has advanced the pediatric cancer field as a pre-declined model for gene and drug discovery. So it's very important benefit for the zebrafish system of pediatric cancer research. The ability to develop rare tumor subtypes in a timely and affordable manner for preclinical drug discovery, as described earlier for leukemias, brain tumor, and sarcomas. In addition, the unique imaging attribute of a zebrafish represent a powerful tool for directly monitoring the tumor cells' behavior during invasion and metastasis, as well as response to the drug treatments. So CRISPR toolbox in a zebrafish. Since it's a first application for site-directed mutagenesis, the CRISPR Cas9 system has revolutionized genome engineering. Here we represented a validated workflow for the generation of a targeted genomic deletions in zebrafish, including design, cloning, and synthesis of a single guide RNAs and Cas9 mRNA, followed by microinjections in the zebrafish embryos and subsequent genotype screening for establishment of mutant line. The versatility and efficiency of pipeline makes the generation of zebrafish model widely used in the functional genetics. So this is the model. So First, we'll separate the male and female zebrafish breeding tanks. Then, using the separating barrier, then we'll prepare a mixture of Cas9 mRNA for each mule of a mixture. We'll add
0.2 ml of phenol red. Then we'll collect one stage cell. Then we are removing the separating barrier within 2 hours after light of fish facilities which are turned on. Then we'll collect it and used it for micro injections. The further conclusion part will be continued by Atif.
Thank you, Neha. So I'm talking about the conclusion part. Zebrafish is an ideal compact complement to existing genetic system. We took this topic why because we have discussed toxicity and cancer model and CRISPR, and this technique was used in many fields like development biology, cancer, toxicology, drug discovery, and molecular genetics. Our aim, why we choose this topic has idea. In Ideathon, we are pitching why because this technique is it's been widely used in the research field. This technique can be introduced in the UG level, where people or student can think out of the box. At least students can think out of the box. And in the field of toxicology, if we introduce toxicology studies in a UG level to study the model, how the environment having environment is reacting with such carcinogenic substances and all, we can easily study using this model. And this is a small scale zebrafish lab. This is above picture. And next picture is a high-tech, large-scale zebrafish lab. And this is the zebrafish husbandry, where different types of fish were allocated in each tank, different types of breeding. And if that any fishes are like insertion or deletion of the genes are there, means we are dedicated to that particular tank. And this is one of the interesting part where fishes are left to mate. Male fishes, usually those fishes after laying, female fishes after laying that eggs will be trapped down on the bottom of that tray. If we left over, if that suppression is not there, then these male fishes start eating those embryo. To trap that embryo, we are using this method. And if we talk about why we are choosing this topic, why because and the thing is, if you come to mice, chimpanzee, as dogs, and Drosophila melanogaster, even these are very difficult to maintain and manage and all. This is a technique where we can utilize their benefit in our research field in order to get skills. What skills in biomedical field or biotechnology field, in genetic and in the genetic field. So I'd like to conclude. Thank you.
The session is open for discussion.
Kaoru sensei, yoroshiku onegaishimasu.
I have questions. So thank you for presentation. So it is not clear for me rule of graphene oxide nanomaterial. I don't know. I don't understand a rule of graphene oxide.
Our intention is like we are only the one topic, particular topic in toxicology, which was graphene, which is graphene. Graphene oxide. How it will utilize in to study the zebrafish model. There are so many other models also there, like lead and asbestos. These chemicals are present in environment, and it can be studied using this model.
Yeah, ma'am. Graphene is we can use even graphene, but the thing is,
it is very costly. Instead of that, we can use graphene oxide, which is less expensive. That is alternative for a graphene, because graphene oxide, which contains a nanomaterial that which produces extreme large quantity through oxidation. So graphene oxide is structurally different from graphene, which also contains oxygen containing functional groups like epoxide, hydroxyls, that which contains structural effects. So here we have here the thought is we can use graphene oxide instead of graphene. There are so many other we can use other different alternatives like formaldehyde, mercury, lead, but the cheap of course is graphene oxide.
So, how much of doxorubicin in graphene oxide? Did you check the content of drug doxorubicin in graphene oxide? So yeah, you are best content of doxorubicin content.
Can you can you ask?
Pardon, ma'am. Can you repeat the question? Pardon, ma'am. Can you repeat?
I see. I want to know the best content of doxorubicin in graphene oxide. So you used a drug delivery system. You used drug delivery system.
Ma'am, we used 50 microgram per ml mg. We are exposed in that.
50
new G. New G per
ml. Per gram.
Yeah.
I see. Thank you very much.
The same question of I am just continuing with the questions of Professor Kaoru. This is the combination of doxorubicin and graphene oxide. Am I right?
Yeah, yes, sir.
If this is the composite, that means you use some reducing or oxidizing agent to prepare the graphene oxide nanomaterial, and you conjugated with doxorubicin.
Yes, sir.
Isn't it?
Yes, sir.
In that is the case, which act as a toxic, whether it is a GO or a doxorubicin.
Graphene oxide.
Okay. You didn't check the toxicity of the DO, doxorubicin?
No, sir.
Okay. This is your UG project or a?
Sir, it's not a UG project. We are just pitching our idea.
Okay. Got it.
It can be used in this way also. Why only we are limiting this technology or this model organism only for the research field or PhD or master's level. We can bring this to UG level where students can open up, where students can think out of the box.
Correct. Got it.
We are just taking on an example. These things are already discussed in the research. We are just bringing up in the UG level.
Great. Yeah. You have any plan to use it right now?
Right now, sir, we are lacking with our department has no that system. In future, we may get. Our sister institution is there, NUC, sir, in Nitte University. There, this technology is there. those people are practicing also.
Okay, okay.
Yeah.
All the best for future endeavors.
Yeah, sir. Thank you so much.
Radha, you.
Thank you for all your presentations. We will now have a virtual walkthrough of the labs and instrumental facilities at the University of Miyazaki. The session will be conducted by Dr. Harish Kumar Madhyastha, Associate Professor, Department of Cardiovascular Physiology, Faculty of Medicine, University of Miyazaki. I call upon Ms. Shannon to briefly introduce Dr. Harish Kumar Madhyastha. Over to you, Shannon.
Dr. Harish Kumar Madhyastha, faculty at Department of Cardiovascular Physiology, University of Miyazaki, Japan, holds 2 PhD degrees. He ignited his research career as a scientist in spirulina biotechnology at MCRC, Chennai. Later on, he pursued postdoctoral research at Miyazaki University that culminated in a faculty position in the Department of Cardiovascular Physiology at the University of Miyazaki from 2006. His current research interests include generation and delivery of nano-sized metallic payloads for regenerative diseases application. His academic credentials are credited with more than 90 SciE indexed publications, H value of 33, carry weight analytic cumulative impact factor of 216.5 and RG score of 35.17 with 6 international patents. His research has been presented in conferences more than 100 times and has been frequently picked up by national and international media. He is also actively involved in many international projects, including ongoing Indo-Japan scientific and academic collaborations. He is Fellow of Biotechnology Research Society of India, Fellow of Royal Biological Society, London. He is an officially recognized Indo-Japan academic spokesperson of University of Miyazaki and engaged in outreach programs to further strengthen the cohesive relationship between Indian academicians and University of Miyazaki, Japan.
We will now be playing the video. The video will also be played offline at Shambhavi Hall.
Faculties of Nitte Engineering College. I am Harish Kumar from University of Miyazaki. This is a southernmost portion of the Japan. Our university name is University of Miyazaki. We have 2 campuses. This is the main campus and this is the Faculty of Engineering, and we have Faculty of Regional Innovation and Agriculture, Arts, Commerce and Sociology, and Veterinary Science, Faculty of Medicine, where my faculty is there, and we have our own helicopter, which is aerial emergency system. We have Central Instrumental Center, which house the almost all state-of-the-art instrumentation, which are mainly used for the biomedical, biotechnological and chemical technologies analysis. This is the main entrance. It is 24/7 open. Immediately after entering the main gate, we have registration. It is online registration. Now we will start the showing the one by one instrumental facility centers which are housed in the different floors of this building. This is the optical labs where major spectrophotometers are housed. One such first one is the FTIR, that means Fourier transformed infrared spectroscopy, a kind of spectroscopy where a sample is irradiated with infrared light and transmitted or reflected light is measured, allowing structural analysis and quantification. Absorbance analysis involves the measuring of amount of absorption of light by molecule in a sample which is wavelength dependent. Infrared light with 2.5 to 2.25 micromolar intensity is absorbed due to the vibration and rotation of the molecules and energy involved. In the infrared spectrum, absorption peaks associated with the functional groups such as OH, NH, SH, COOH appears at specific wave numbers. These regions are referred as characteristic absorption bands, and this space can be analyzed in order to estimate the structure of the compound. This is photoilluminance spectroscopy, which is routinely used for studying the structural changes in the conjugated systems, aromatic molecules with rigid planar compounds due to the alternation in the temperature, pH, ionic strength, solvent degree, and ligands. A single fluoroprobe can generate thousands of detectable photons. And this is the Raman spectroscopy, is a popular technique for the analysis of the molecular structure, and it's considered the complementary to the infrared spectroscopy. Inventor of this great instrument, still charming instrument, is Professor C.V. Raman, our own Indians from Indian Institute of Science. Way back long back, for that he got Nobel Prize. Raman spectroscopy is based on the Raman effect, which was first identified by the Chandrasekhara Venkata Raman in 1928. Please note 1928. The Raman effect is based on the scattering of light, which includes both elastic, or in scientific time it is a Rayleigh light, scattering at the same wavelength as the incident light, and inelastic, that means Raman scattering at the different wavelength due to the molecular vibration. This instrument is extensively used to identify the peak wave absorption spectra of the compound or a molecules which has the vibrational molecular structures. It detects with the various lenses and can be analyzed with the microscope. This is XRD, that means X-ray diffractometers, X-ray powder diffraction spectroscopy, which has the three basic elements: one, X-ray tube; second, sample holder; and X-ray detector. When electrons have sufficient energy to dislodge inner shell electrons of a target material, characteristic X-ray spectra are produced. These spectra consist of several components which are mostly generated by K alpha and K beta system. X-ray powder diffraction is most widely used for the identification of crystalline material. Please note, it is used for the crystalline materials like minerals, inorganic compounds, and nanomaterials. Determination of unknown solid is a critical to study the nature of the geological components, materials, environmental science, and engineering, including the biology. One is X-ray gun, sample holder is in the black color, and stick to the sample in the sample holder and start analyzing with X-ray bombardment, which is on the left side of the material, left side of the instrument, and mirror, which is the X-ray detector, is in the right side. This instrument belongs to the Pananalytical instrument company, the most advanced one, and we have database for the all materials. Then we will analyze the material component. This is the TEM, transfer electron microscopic lab, whereas ultra thin sections of the material or a biological tissue will be bombarded with the X-ray, and bombarded X-ray will be transferred through the material, and you get the image through this instrument. It is called TEM. Of course, this is very old instrument. We first installed this instrument in 1962. It's a very old instrument, but still working. And image can be captured and analyzed with KinView system, which is installed below the instrument, and finally analyzed with the computer software. Another important aspect is this instrument is attached with the IDAX for the elemental analysis. Electron density analysis by X-ray, IDAX. This is the analytical section. This is the whole instrumental facility setup. And we have SEM, scanning. This is the specimen preparation room. In order to prepare the ultra thin sectioning of the material, we should first cut the material through the microtome by using different knives which are made of the glass or diamond, which is the blue color is the diamond one, the triangular glass material, the yellow and white is the glass knives. It is once again cutting is through the microscope, ordinary microscope, and we have automatic ultra thin sectioning of the material is also available, which is controlled by the computer. Need not to use any human interference. And these are all the machine to prepare the glass knife. We have SEM. It is called scanning electron microscope. It's a type of electron microscope that scans the surface of the living or non-living objects that uses the beam of electrons, which is moving at low energy, to focus and scan the specimens. The development of electron microscope was due to the inefficiency of the wavelength of the light microscope. In the light microscope, we can get the maximum enhancement of the object up to 100 times. Whereas the scanning and electron TEMs, SEM TEM, we can have up to 1 million magnifications, huge magnifications. Electron microscope have very short wavelength in the comparison to the light microscopes, which enable the better resolution power. Right? The secondary electrons are emitted from the specimen, play the primary role in detecting the morphology and topology of the specimen, while the backscattered, which are housed in this chamber, shows the contrast in the composition of the element of the specimen. The black color is the sample loader, and once again it has the EDAX attachment to analyze the elemental compositions.
Hello, audible, sir? Hello, sir, audible? Oh, yes, sir. We'll go ahead, sir.
It is now time to announce the winners of the Ideathon. I request Dr. Venkatesh Kamath to please announce the winners of the Ideathon.
For the Ideathon event, we had 3 teams, Team 1, Team 2, and Team 3. The final results are here. Team 1, consisting of Anjali and Pratiksha, scored first prize. I mean, they bagged first prize. Anjali and Pratiksha, Team 1. Then Team 2, Rashmita and Anushri, they bagged second prize. And the team number 3, comprising Neha Salyan and Atif, bagged prize number 3, consolation prize. Over to Athira.
The teams will receive the award certificate shortly. Melody Beater once said, "Gratitude makes sense of our past, brings peace for today, and creates a vision for tomorrow. It turns denial into acceptance, chaos to order, confusion to clarity." On this note, I would like to call upon Ms. Nikita to deliver the vote of thanks. Over to you, Nikita.
Good evening, one and all. It is my pleasure to extend gratitude towards each and every one of you who has attended and contributed in making this program a successful one. I would like to thank the invited speakers of today's event, Dr. Kaoru Ohe, Dr. Betasivi Purbasari, Professor Dr. Praveen Kumar Shetty, Dr. Shyam Prasad Sajankila, Dr. Radha Madhyastha, Dr. Harish Kumar Madhyastha, and Dr. Yuichi Nakajima. Thank you for taking the time out of your busy schedules and bearing with the time difference to share your insights and knowledge on biophysical techniques for biomedical research skill improvements. I would like to thank the seminar chair, Professor Dr. Nozomi Watanabe, Organizing Secretary Dr. H. Venkatesh Kamath, the conveners Dr. Ujjwal P. and Dr. Harish Kumar Madhyastha, the Faculty of Medicine at University of Miyazaki, Japan, and the faculty at Department of Biotechnology, NMAM Institute of Technology, for organizing and facilitating the students to make use of this opportunity and gain more knowledge. I would like to thank all the student volunteers here at NMAM Institute of Technology for helping out with the organizing and live streaming of this event on YouTube. I would also like to thank all the students who have participated in the Ideathon. Special thanks to the sponsors of this program, Japan Science and Technology Agency. Last but not the least, I would like to thank all the audience who have been with us throughout the event. Thank you, one and all.
Thank you all my student volunteers who worked as per the instruction. In fact, beyond the instruction, with the proper timing, matching with the timing requirements or any other kind of technical glitches that they could solve in time. Thanks to all the volunteers, and a special thanks to University of Miyazaki and Professor Harish Kumar Madhyastha sir and Radha madam for and department faculty of medicine, Dr. Betasavi or Dr. Purvasiri for giving wonderful lecture sessions. Special thanks to Harish sir for giving us an opportunity, giving an opportunity to our student to virtually visit Faculty of Medicine at University of Miyazaki. Special thanks to you, sir. Yeah, the program has come to an end, sir. Over to you, Harish sir, if any few words. Sir, not audible, sir. Oh. Yeah, sir. So, network issue or something. Because of that, he's not audible. He's conveying his thanks to all the team members, faculty at Department of Biotechnology, N.M.A.M Technology, and guest speakers. It was a wonderful event and a good interaction. He's conveying his thanks through the message. Yeah, thank you also. Thank you. Shall we log out, sir, now? Yeah, thank you, sir. Bye, sir. Have a good day.